My name is Elizabeth Bradshaw and I'm an instructor at the Center for Neurologic Diseases in the Department of Neurology at Brigham and Women's Hospital. I am focused on studying the innate immune cell type monocytes in human disease. One of the diseases we're examining monocytes in is Alzheimer's disease. Recently, the CD33 locus was identified as a susceptibility locus for Alzheimer's disease. CD33 is a known myeloid marker. It's also highly expressed on monocytes. Therefore, we would predict that CD33 locus may influence the function of monocytes. We demonstrated that this genetic locus influences the surface protein expression of CD33, in that people with the risk allele have higher CD33 protein expression on their monocytes than people with the protective allele. We also found that this common genetic loci was associated with reduced phagocytic ability of monocytes, in that subjects with the risk allele had reduced ability to uptake amyloid beta compared to people with the protective allele. If the monocytes have reduced phagocytic ability, we hypothesize that perhaps the genotype correlates with the amount of amyloid beta deposited in the brain. We utilized 3 longitudinal studies in which they examined the amount of amyloid deposits in the brains, and we found that subjects with the risk allele had a higher chance of having amyloid deposits in their brain. This study is so exciting because we are able to start with a GWAS-associated susceptibility locus that is only weakly correlated to Alzheimer's disease susceptibility, and find that it has a strong association to a molecular phenotype in monocytes, such as uptake of amyloid beta and CD33 protein expression. And in addition, we are able to correlate it with amyloid beta deposits in the brain. Hopefully our work can be used for drug development, in that either it can identify new targets for therapeutic treatment, or perhaps it will help identify certain subsets of subjects that will respond to certain treatments.